we're going to review a little bit of what we learned on Wednesday so that it's a good recap for everyone. Anyone who was here, do you want to say what your main idea from Wednesday was? Where are the random effects stored usually? In the variance covariance matrix, exactly. That means that they don't affect the expected value, right? And we just add them because we know they, especially in a design experiments setting, we add them because they are some components of the variance. We know where that part of the variance comes from, and so we want to account for it. But it's not really a treatment that we really thought through. It's just, for example, a block, or we're going to see other structures in design experiments today, right? So for them all, we usually write out so remember, y is a vector. It basically has, let's call it 3 components. These would be the fixed components, the random components, and the residual. This was the model equation form, remember? And then if we want to write it out with the probability distribution form, we would say y is normal. This is mu, right? This is the expected value. And the variance covariance matrix is the random component, right? Plus the residual component. And this G matrix here, and I'm writing here G and R, because afterwards, when we study repeated measures, for example, you can, and it's not something we're going to go into detail, but you might read about this somewhere sometime, about G side and R side covariance structures. And this is where they come from, right? R side are in the residuals. G side are random effects, okay? And they all are part of the variance covariance matrix. So, quick recap. What was the difference between fixed and random? We said this we expect there's like one true value out there that we want to find out, that we want to study. It's usually very thought through treatments, for example, carefully selected. And this is usually some component of design that we add to them all because it explains some part of the variance, right? Here we estimate the beta hats, right? Remember when we put hats on things, that means it's an estimator, right? And here we estimate the variance. Or this is our estimand, let's call it like that. This is what we are trying to estimate, beta, and here we're trying to estimate the variance component of the block, for example. And so this has huge implications for the degrees of freedom, and if we waste a lot of degrees of freedom, sometimes it wouldn't even make sense to put them all random, right? And this is going to make sense in a minute. Questions? So today we're going to review a few of the most common design experiments, at least in my experience, most common. If someone else disagrees, feel free to jump in, and because this is very field specific, whatever design is more common, right? So really quick, design experiments. I put this discussion on blocks, fixed or random, because I think it's very interesting, and it makes you think through a lot of what are we doing actually when we fit our fixed effect versus a random effect, right? And then a few examples with applying these concepts that we reviewed. So really quick, everyone here took 720 or is familiar with design experiments? Yes? Okay, most people, yes. Okay, so then you can tell me why it's important to have, for example, randomization. Like if we have a field that we can divide in many plots, we don't just say like, this is gonna be treatment 1, 2, 3, 4, 5, 6, 7. We randomize what goes in what, right? Do you remember why that is? Yeah. It basically allows us to make that independence assumption that then avoids any type of biases, right? Now, remember that usually we say we add random effects when the observations aren't independent anymore, right? So for example, the most common type of statistical model, like if you run least squares, OLS, right, which you can do everywhere and most people do, it's just whatever predictors you have here and the error, right? No random effects. That means that this, we can call this V. This V is just sigma squared times a diagonal of 1s, right? And these are zeros. That means that this is the variance and all observations are independent because these are zeros, right? Because they are independent, we put zeros here. So, okay, that's that. Then replication, we need to have good estimates of the variance and to know what the uncertainty is, right? And local control is basically to be comparing things that are in similar conditions, and if not, to correct for those similar conditions. Example, blocks, right? There is one area in the field that is more fertile. We want to make that a block, right? And say all these plots were in similar conditions. Okay? Experimental units versus observational units. That's a big deal for animal people, right? So, what was the experimental unit?
The unit at which treatment is.
Yeah. So, if we are randomly assigning treatments to these plots, for example, each plot is one experimental unit. If I sample many plants in that plot, or if this was like a pen and has many animals, each individual, if I observe them all, they are subsamples. But the treatment was independently applied to the whole group of plants, of animals, whatever. Okay? And when we don't account for that, let's say that we have 3 observations from 3 different plants or from 3 different animals, right? If we don't account for the fact that they are actually subsamples, if we don't add that to them all will automatically, it will be implied that they are actually true replications. That will make us overconfident about how much signal there is, right? Okay, and so that's the basic things those are the basic things we need to know about design experiments. And now we're gonna see four different types of design experiments and how that will affect the independence or not between observations, and how that translates to the model, and later on to an application, so the model, the code, the interpretation, okay? So, completely randomized design, and further down you will see kind of little maps, examples for agricultural experiments, so like plants, basically. And there are 2 boxes for animal and like kind of lab settings. Feel free to tell me if something doesn't make sense. But basically, the whole idea for the CRD, completely randomized design, we can assume that in a field setting, the whole area is approximately homogeneous, right? So if I grow a plant here or here, they're going to experience similar conditions. So there's no need to control for any higher or lower fertility whatsoever, okay? And so what we do, we go and we apply the treatments randomly. The treatment structure doesn't matter. It could be just one treatment factor, or it could be like a factorial treatment structure. Doesn't matter. We're going to combine them all and apply the different treatments to a field. Okay? So, because this is approximately similar, we can assume that they are all the observations, since they were independently assigned the treatments. If we have one observation per plot, each observation is going to be independent? Yes? So, how would the stat model look like? It's over there, so I'm going to show the drawing. Okay, that's that. Let's start from scratch. If we have 8 levels for the treatment, okay, 3 reps, Y ith treatment and jth repetition. Overall mean plus what? Treatment effect of the ith treatment. Anything structural that we want to add, how are the data generated? Completely independently, right? So that's just the error, right? Of the ith treatment, jth repetition, okay? I is 1, 2, 8, j is 1, 2, 3, okay? Variance covariance matrix looks like this. Sigma squared in the off diagonals, because what's missing here is the distribution of the residuals, right? And all these are zeros. Right? Because we are assuming independent observations. let's say that the field is actually not so homogeneous. There are, let's say, 3 distinct areas that are homogeneous inside, right, but are different to each other. So this will change a little bit. We're going to have the same plot, the same field, but we could divide it into 3 homogeneous areas, which are called blocks. Example in animal trials are probably repetitions that you run, right? Once, twice, 3 times, or different rooms, right, where you have your animals. Microbiology, you could be running the same experiment in several, let's say 1 week you do it, then you repeat it another week, then the next one another time, right? Now, we say that these are all similar to each other, like they share the block, right? So the rest is the same. But how are the data generated? It's good to think it in that. So first, how are the data generated, and then the treatments, then the fixed effects. First of all, we have 3 different groups, right? And then inside each group, it's random again, right? Inside each group, they are independent. But we have these 3 different groups. So in this part, we're going to add a random intercept, okay, for the jth block, which is like the jth rep, right? And still the treatment is the same. Treatment effect is the same. How does this look like? Let's say that the first 3 observations are from block 1, and these are from block 2. This gets a little bit more tricky. First of all, what's the distribution of U? J is normal, mean 0, variance, sigma U. Okay? And so this will go to this is going to be sigma squared plus sigma U, right? For all the diagonals. But now also, for cases where we share the block, these are correlated. But you see that the mean, which would be this, the expected value of y, doesn't change. Right? And in fact, the estimator of beta, so of this, is unbiased, right? You'll get the same estimate if you keep the blocks or if you don't keep the blocks, as long as it's balanced and everything, right? And randomized complete block design. You're going to get the same estimate. But what happens? Whatever you don't explain is going to go to the error. And if your error is too high, and all the point of the experiment was to make mean comparisons, you probably will not detect any difference between anything, right? Because the error is so big. The estimates are going to be the same. Now, if you know where the variance comes from, you can add it to the model, improve your model, have more precise estimates, right? Because that's what happens when you have a huge model that you're not explaining, all your confidence intervals for your means are super wide, right? Now, if you can say some part of the variation comes from the blocks, some part of the variation later on comes from the whole plots or from correlation in time repeated measures, then you can more precisely estimate your effects or your means. Now, any questions so far? This is where we have our discussion on blocks fixed versus random. Okay? Anyone wants to say any comments, what they do? So, if we fit our blocks as fixed, it would be basically something like this, right? Blocks fixed. Overall mean, treatment effect. I'm going to put alpha better. Effect of the block and the error. So here, remember that when we are estimating fixed effects, we are eating up degrees of freedom, right? Each new fixed effect is a degree of freedom. Versus here, we're just estimating this variance, right? That's that. Now, I think it's an interesting discussion because especially in field experiments, if you blocked really well and your blocks are really different, your variance might be pretty high, right? And this is for field experiments, right? So if you really have a more fertile area and one less fertile area and one intermediate, first of all, we're estimating variances with 3 points, right? Because we have 3 blocks. And then our variance might be huge. That's not going to change our inference because I'm going to show you now, but this variance doesn't go when we do mean comparisons, right? The standard error of a mean difference only takes into account this variance. So it's not going to change our inference, but it's something to consider. Have you ever seen this case? I'm going to show you what, whoops. This is showing, this is from a paper that I highly recommend. This is showing control, treatment A, treatment B, mean comparisons, model with fixed effects, blocks as fixed, model with blocks as random. In both cases, we say that they are different with p less than 0.0001. But if you look at this plot right here, you see very wide standard errors, right? The lines are standard errors. And it's not that it's wrong, but some people, and this is just like FYI, I have seen this and heard this as comments in a lot of presentations and whatnot, and people showing plots like this saying, yes, we saw statistically significant differences, and people asking like, yeah, but they don't seem significant. And that's because here we're showing the standard error of the mean that considers the the variance of the error and the variance of the blocks. But when we do mean comparisons, we don't use the variance of the blocks. So this makes sense. But you have to know where this all comes from. Does that make sense? So You see the first column is for blocks as fixed, and the second is for blocks as random. Okay? The point estimates as we are the same, right? The mean estimation doesn't matter, right? What happens is this when the blocks are random, that variance goes to the standard error of the treatment means, right? Not to a comparison, right? This is the same. So our inference is going to be the same. But when we show it, it's good that we know where our standard errors come from. Because if I got that question as a master's student, I wouldn't have known the answer, to be honest. So it's good to be mindful about what we are estimating and what we are modeling. And most likely, in this case, this was a field experiment, probably whoever performed that experiment really knew the field, because apparently the blocks were actually different, right? And that's why we get that variance so high. Yeah?
So should we not report SEMs if the interpretation of them can be a little bit challenging? Or there's a lot of overlap, but the hypothesis testing says there's a difference. And
Yeah.
the interpretation's there.
Yeah.
So should we not use SEMs? Or confidence intervals better? Or
I usually use confidence intervals, but the confidence interval is going to come from the same standard error, so it doesn't matter. I do highly recommend reporting some kind of uncertainty estimate, because honestly, if I was looking at this with no standard errors, it's hard to believe. I don't know, and it's hard to know what it implies. And again, as long as you report your model, ideally we will report variance estimates as well, and that would be the end of the story. It's not very common in most applied fields. Yeah. Does this make sense to all of you? I wanted to show you all this because this is very common, and that's a very common question. Like, are you actually seeing significant differences? Because I can't see that in your plot. And that's probably because you were using random effects for the blocks, for years, whatever, right? And for that, I also recommend that paper, Dixon's 2016. Then we have this, the fixed effects are more expensive in terms of degrees of freedom. You really don't feel it when you're doing an RCBD, but you will start to feel that loss of degree of freedom if you do a split plot analysis with only fixed effects. We'll see that in a minute. Any comments, questions, concerns? You wouldn't imagine the discussions statisticians have over blocks fixed as random. Like huge debates, seriously. There was this ACSTATS conference that is still held, but now it's ambulatory. It's 1 year in a different university. But it used to be held here for decades, and there were a lot of discussions among the applied statisticians on blocks, either if they should be modeled as fixed or random. So you see that both positions are okay as long as you know what you're doing, right? It's not that one's wrong or right. As long as you can justify why you're doing one of them, that's okay. And this paper actually comes from a discussion in one of those meetings. So, questions? So, what we know is that, what were we, yeah?
So, based on the two plots that you showed in our discussion
Uh-huh.
How much the use of a random slope instead, especially when you see that if you increase so much your SEM would be a better approach rather than a random intercept or the
A random slope? Well, in this case, we don't really have slopes as such. Because I don't know if you can read from there, but it's control treatment A, treatment B. So it's like, it's this case where we have an overall mean, and then, and how this would look like. This is control, this is treatment A, and this is treatment B. And this is whatever response we got. And so it's not that this is continuous, right? The predictor is categorical. We have 3 categories. And so for control, we have like the observations around there, here around here, and this one's around here, right? And so what the models are doing are basically, we have an overall mean, that's the average across all observations. And then we have some treatment effect for a control, some treatment effect for treatment A, and some treatment effect for treatment B, okay? And then within those, and each one of these is unique, right? Because this would be, this right here would be tau 1, this would be tau 2, tau 3, right? And then within, like after you added the overall mean, the treatment effect, this, then you're gonna say, okay, I hope you can see from there. In a random effect scenario, you see, okay, centered at this, at the expected value, we're gonna see a small normal distribution, oops, centered at the expected value because the expected value of the random effect is 0. And then, and it's the same distribution for all, same variance for all because it's the variance of the blocks. And we say, okay, block was a little bit lower for all. And that was the random effect of the block. Block 1. Block 2 was here. Block 3 was here. Okay? And then the error. If you had a fixed effect small, but you see this is like on the intercept that where you're only shifting up or down. You're not shifting any slope. You're shifting everything up, all the blocks 1 up or down, all the blocks 2 up or down. Does that make sense? But back to your question. Oh, yeah. Another thing, so this is more model specification. Since we have a categorical predictor, our random effect is going to be on the slope. It could be on the effect, but that's not usually what, in design experiments, we assume that the blocks don't have any type of interaction with the treatments. So a slope would be the effect of a treatment. This is the effect of a treatment. We only put random effects on the intercept. That's why the other day and today also, we're going to see when we program that in R, we put 1 and then whatever block, whatever random effect we have. 1 is, it means it's acting on the intercept. And for design experiments, we're going to keep it like that. If you have observational data or your animal science or VetMed, well, if you were studying a group of animals and you're measuring their weights through time, it really depends on the setting. you could use either repeated measures, or we're going to see that after, or a random slope. Otherwise, in design experiments, I don't think, you usually assume that the design is a design, and that goes as a random effect on the intercept, and it's not affecting the treatments. It's just describing how the data were generated. All good? OK, so actually, that's that. So each block, they are different. They're different blocks, right? They're different to each other, but they're homogeneous inside. So inside, we have this random allocation, independence assumption inside the block, not among blocks, right? That's why, Now, for split plots, we're going to keep the same drawing and add one thing. Split plots, usually, we see them when we have at least 2 treatment factors. And usually, one of them has is either hard to apply. Let's say fertilizers are usually have a minimum area from a machinery perspective, right? And so we have to make the experimental unit, so the minimal size we can apply and treatment independently is pretty large, right? Fertilizers, irrigation, or sometimes we can fit, like we have some type of lab equipment that only fits so many samples, and that's everything we can use, right? Sometimes we have practical restrictions. And so what we do is the randomization process happens rather sequentially, right? So what happens is this. Let's say that we have 2 treatment factors. Now for split plot. Factor 1 with 4 levels, and factor 2 with 2 levels. And it's still at 4 by 2 factorial, right? So we have 3 blocks. This is a split plot in an RCVD because we have these 3 repetitions, right? Then, first we randomize the first factor. We randomly assign this experimental unit. This is now the experimental unit for factor 1. To all of this, we apply, let's say, treatment B. Here we apply treatment D. Here A, and here C. And then, so these are the experimental units, right? Could be, for example, a pen of so many animals, right? And then we split that. Let's imagine in a field, we split that plot into 2 and apply that other treatment. Let's say this will be 2. This will be 2, and this will be 1. 1, 2, 1, 2. This is random. Which one goes to which? Okay? And so you see that we randomize the blue ones, so the second factor, inside the other one. So this is like a mini block, right? When we are applying the blue treatment, we are randomizing inside this, just like before we were randomized inside the block. Does that make sense? So it goes blocks, right? The observations are blocks. And inside here, it's not that everything is independent, right? We have like small blocks again inside the other blocks. Does that make sense? So now we're going to change this part because now we have 2 treatment factors that we're studying. But first, let's study this. We have first the block effect, right? Then the mini block effect. We could call it mini block, whole plot in actual experimental design terms. Whole plot is this. Split plot is a small one, right? So the whole plot would be, this is the same, distribution is the same. Whole plot would be if this is I, j, k, this is factor 1, j is factor 2, and this is the repetition. The kth block. The ith. Now we're referencing the mini blocks, right? So this is, for example, the name of that mini block. This would be B, D, A, or C, right? And these are levels of this factor. So this is the random effect of the ith whole plot inside the kth, or with parentheses, however you like, inside the kth block, because it's this treatment and this block. So we know it's this one. Okay? And then, now inside this was randomized, so it's the error, the independent error, again, right? But now we have, like, hierarchically many sort of blocks. Because if we're comparing only factor 2 in this block, these are like the blocks for the second factor, right? Because we have it repeated 4 times. If you forget about these letters, this is like an RCBD, right? It's like an RCBD win, right? So we have here the errors. This is also normal. And the error as well. And how about the expected value? Overall mean, treatment factor 1, treatment factor 2, and their interaction. And I use Greek letters, but if you want to use letters, as long as it's understandable, it's all good, right? As long as it's understandable. Questions? I'm not going to write. So to this, we would add the variance of the whole plots, and then everything, all the observations that share whole plots also share that sigma squared. You see? So this means all these 10 observations, and there's a data example above this. The idea of the web page is that you can have an accessible way to still access whatever we learn here. I lose all my papers, so that's why having a website is very convenient often. Diagonals, right, the sum of all the variances, and then the sum of these apparently share the block and the whole plot, right? And these only share the block. Okay? And in the end, we're only doing this because we want more precise estimates. And if we describe how the data were generated, that's usually more convenient to get those. Now, the last design we're going to see before we go to a break is repeated measures, right? Which is very similar to a split plot. Only one thing changes. You see that in this example I put here, we had 4 treatments in the 4 levels in the second treatment factor. And remember that within each whole plot, we randomly assign which one went to what, right? Like, we randomly assign the levels of the second treatment factor inside here. A repeated measures design is very similar, but the second treatment factor is time. And time cannot be randomly allocated because time is unidirectional. It happens. Entropy grows, right? And so it's very similar. But what we see is that now the four levels, instead of being physically divided, they are temporally divided. And that's why it's not random anymore, right? So we have to account for, if it's not random, you know that we said, okay, these are IID residuals because inside here we could randomly allocate, right? So we can assume that independence. If we cannot randomly allocate, we cannot make that independence assumption. Or we should not. We can test it. Does that make sense? Yeah? And so there are 2 ways of accounting for that. You see, we're going to keep this because it's treatment factor 1. Now treatment factor 2 is time, but we still have a time by treatment interaction, right? This is going to change. And we still have this block. And this is the whole plot random effect. It's going to change a little bit, right? And we're going to say this. You see that this subscript and this subscript are the same. This is still going to be sigma squared I, right? Sigma squared in the diagonal, zeros in the off diagonals. This is going to be no zeros in the off diagonals because that's where we're going to include that dependence assumption. So let's do J is time, right? It's a jth time. So that's why I didn't include J here, because we're going to show the different J's in the elements. This is 4. So we're going to have just like before, sigma squared V, right, of the whole plot. And now in the off diagonals, this is an R1 autoregressive 1 correlation function. And that means this is the Greek letter rho, rho, like that. I'm going to show you what this means in a second. And this would be time 1, time 2, time 3, time 4, assuming they are about regularly spaced. The variance is sigma squared v. And then as you go through time, they are correlated, but they are less correlated every time you go further away in time. We're going to have a break in 5 minutes. Okay, this is before we used to have, let me see. Yeah, we used to have zeros in the off diagonals. But right now we're saying that the different times are actually, today is similar to tomorrow, and it's going to be similar to the day after tomorrow, but a little bit less similar, right? And that's how this, since we're doing rho, then rho squared, then rho cubed, right? It's going to decrease with time. Making sense? Okay. And so I'm not going to go into the details of this, but you see that this is a random effect, right? This is still a random effect. So that means that like this is still acting in here. So this is one type of G-side covariance, right? Which really doesn't matter if the response is normal. It starts being more important when the response is not normal. More details, messy data 870. Just so you know, if you did this for, instead of the random effect for the y's, so this would be the residual. Because right now you're saying that so like when this is v, y is for the same is sigma squared and a diagonal of 1s and zeros in the off diagonals, right? Like this, it's G side. But if this was the case, if you added it to the residual structure instead of the random effect structure, it would be R side. This is not really important for this more introductory class. But just to keep in mind. In the end, for normal, it doesn't really affect inference. So it's the same. Because they act on the same level, we don't really care. But then, if you have other types of applications, you might want to read more about conditional versus marginal models. In the end, we're only solving for a split plot, sort of, where we can't randomize the second treatment factor, right? And that's why we include this. Because since we can't randomize, they're not independent, so we got to account for that. Questions? The next part is applied examples. I would give you guys 10 minutes to freshen up, go to a restroom. Because how aren't they confounded? And I guess this is in a G-side modeling scenario, right? We could also model the residual as a sum of these two, because this is describing how, describing the intrinsic variability, let's call it. And this is describing for a given treatment in a given block, that means one experimental unit, right? We will have this correlation through time. right? It's going to be a baseline, and then as time passes, the correlation's going to be smaller and smaller. And with this, we only estimate the variance and this rho, right? This R1 coefficient. And this rho is usually less than 1, and so as you multiply something that's less than 1 squared, well, this would be squared or to a third, it's going to reduce that number, right? And correlation is going to get smaller and smaller as time goes, right? And this IK is basically just identifying one experimental unit, because the whole point of repeated measures is that you are measuring the same experimental unit through time. And so the point today is going to be very, it's going to be associated to the point to the same observation tomorrow, the day after, but that covariance, that correlation is going to get smaller from today until like a week from now. It's going to be smaller than from today to tomorrow. Does that make sense? Yeah.
So let's say this there is some cyclicity, then does this correlation keep on decreasing or it will increase also?
Sorry, can you repeat that?
If there is some cyclicity or diurnal nature, then I think it can increase also.
If there is, yeah. So good question. If there's like some circadian rhythm or something like that, where it increases, decreases, and it's not something that, yeah, it might go, I'm thinking it might be more a fixed effect than a random effect, first of all. Or my approach would be to model it with a different tool that would be splines, and you can make splines that are that have some regular function of time, for example. But that's already like non-parametric methods and would be for another workshop. But yeah, this is just saying today is similar to tomorrow, and to the day after tomorrow, but a little bit less, and the day after, but a little bit less, and so on. Okay, yeah?
How does, if you had that with subsampling, is that measurement on that animal, or how does, if you add on subsampling on top of that?
Yeah, yeah, that's why, very good question. I wanted to get to that. Same experimental unit, right? So any animal, if we have a pen of 5 animals, it's still the same experimental unit, so it still would be repeated measures, right? If we're always measuring 3 pigs, and we're always measuring pig 1, pig 2, pig 3, we have subsampling and repeated measures, right? And then the best way is to follow that pig through time, each pig, but also account for the fact that you are having each progression in time is not an independent sample. They are related to each other, right? But that's it. As long as you account for it too, it's all good. Yeah. Is that okay? We all good with this? So, let's say that for all cases we have 2 treatment factors. We're estimating the main effect of each, their interaction. Completely randomized design, right? Everything is independent, so we only have the residual. Randomized complete block design. Oh, sorry. Right? The residual and the block effect. Split plot design would be without the J, right? I is the whole plot, and it's inside each block, right? 2 things to note here. All the VIKs arise from the same distribution, right? Meaning each mini block arises from one distribution, right? And lastly, the repeated measures, where we follow 1 through time. And now we have to include J because J is actually not independently allocated, and that's why we have to model that non-independence, that dependence through time. One last thing before we go to the examples. Very often, I don't know about other fields. In agronomy, it's very often we want to include environmental covariates to explain more of our variability, right? Of yield and whatnot. And that's fine. But it's just a thought that I didn't know when I started studying statistics. That's why I'm saying it. Note that always we have, and this might sound very obvious once you're already very into mixed models, but you see that we have UK, IJK. These subscripts are always discrete values, right? It's 1, 2, 3. They are levels of things. They are, right, categorical variables. So even here, time is categorical, you see? So including, for example, nitrogen or temperature or, I don't know, rainfall as a random effect would only categorize it, right? Because there's no continuous variable that would go into this, at least in my imagination. If you have, we can write a paper if you have an idea, I don't know. But you see? It's just how to think about mixed models. It's only saying what is similar to what. Could be years, right? Years that are similar to each other. Questions?
Whereas covariates are numerical variables.
Yeah.
Treated as basic.
Yes. Yeah. Exactly. But I feel, and this is again my personal opinion, a lot of times in life sciences we approach statistical model first programming and then figuring out what that code was doing, right? Ideally, we want to do it otherwise. First, like, okay, what's the best statistical model I can fit to these data? And then, okay, how do I program it? And will it actually converge, right? But if you go that way, it's probably more pain-free. More pain-free learning process. Okay. And now we have 4 examples. If you click here, you should be able to download the code. And I'm going to open our studio. And we're going to do it a little bit more live. So that we can really inspect the objects. I didn't want to put them on the website because it would be a huge mess of code with no explanation whatsoever. If you don't want to follow, like, code along, I will load this to the website once we're done. So you don't have to copy anything. You can just focus on listening and seeing what we're doing. Can you all read that? Yeah? Okay. Okay, so really quick, what I like to do when I work with R is, first of all, in my project folder, I like to have folders with things. One folder with data, one folder with scripts, and usually one folder with outputs. would be like this. This is not my computer, so it's not very nice. And this is where figures go, or tables, right? Okay. Now, and I have one here. If you look in the upper right corner, this is one project, and you want to have one project in R for each project, or RStudio, for each project you have in real life. If you're studying the different feed treatments for swine production, that's one project. And then when you're studying antibiotics, that's another study, that's another project in R, okay? In my case, when I was doing my PhD, I had one project for each chapter of my PhD, if that makes sense. And one project for each class I was taking. So each course in general, not one for each new class. I actually started doing that at first and then realized it was nonsense, okay? And then when you do your scripts, a good practice I like personally is to load the packages first and then start. I'm gonna restart this. Okay, so we're gonna load these packages. This options function is setting the contrast to 0, so meaning everything will be compared to that overall mean. And if you don't put this in R, you're gonna get a different thing. This is not an R class. We can discuss this later. But it's gonna be easier to interpret this way. So these data come from this package, agridat, that is a bunch of data from agricultural papers that is usually very useful to teach some concepts of good design experiments and design experiments that went sideways and how you can fix a lot of things, like spatial effects in randomized complete block designs. In this case, we have a sorghum experiment that was actually multiple environments, but we're going to filter only one, so keep only one, because each environment had one RCBD. And right now, we're going to have one example with an RCBD, okay? So what we do, remember for the RCBD, we have overall mean, treatment effect, that is the genotype, and then the random effect for rep, this, and nothing else, right? So, in general, remember that we have y is x beta times zb plus zb plus the error, or zu, right? Fixed, random, and this is a more general way of writing it. In this case, and this in R is whatever response tilde, our fixed effects. And for design experiments, we're going to put a 1 because it's going to be a random effect on the intercept only. Vertical bar, random effects. The assumption we were doing on Wednesday and today is that this is normal, right? And let's call, okay? And that in GLEM-TMB, we're using a lot of default things. But for example, here we're actually saying that the family is Gaussian, right? with an identity link function. Okay. Once we fit this model to the data, which ideally we would do exploratory data analysis first, check for that the data look fine, right? There's just not enough time today to do that. But ideally you would do, let's say, a simple plot showing I like doing this. Oh. I like doing plots like this. And just this way, you see that there's no nonsense values, right? Everything's relatively reasonable. There's no typo putting like 1,000,000, right? Everything seems fine. And we really can say, we will fit them all. These data were designed for. So there was one design experiment, so that automatically gives us the model we should fit. And then we do model diagnostics, and then if something fails, we can go and see what we should fix, right? But usually when we have a design experiment, that design experiment is associated to a certain type of model, which we studied the hour before, right? And so the package Dharma, which I made you download, has a very useful function that simulates residuals. For normally distributed data, again, you can use whatever approach you like the best. The thing is that for non-normal data, it's much more difficult to judge if a model fits the data well or not. Because what we will see on Monday is that the whole concept of residuals is very different, because for binomial data, for example, there's not an assumption of a distribution for the residuals, right? Right here it is, it's normal. But for beta, binomial, Poisson, there's not an assumption for the residuals. That's why it's much harder to check if the residuals look fine or not, and which ones look better than the others. So I don't know if you know Ben Bolger, but this is a guy who at least lives in Canada. I think he's Canadian. He's behind the LME4 package, the GlimTMB package, and he is also behind this Dharma package. He and a lot of other people, obviously. And this basically simulates residuals according to the model that's been fit, so this RCBD model, and gives you a lot of help to determine if it fits the data well or not. So if you have over dispersion or heteroscedasticity, right? Those are things that you will see in these plots. So you get, whoops, let me see. One. There you go. Well, it's not so nice, but it's okay. So you get a QQ plot to see if the residuals, these are scale residuals, right? And simulated residuals, simulated with the model that was fit and compared to the residuals that you actually observed. And you get for almost free a bunch of tests to test if the distribution is the one you assumed, and if the quantiles of the residuals are approximately okay throughout the predictor. We'll discuss this more in detail on Monday. For now, just believe me, you're gonna have to believe me that this shows a relatively good fit. And again, all models are wrong, right? It's just we have to fit something that's not super bad for our data, and also not bad. It should make sense. It should be a reasonable set of assumptions for our data. But in the end, all these are type 2 errors, right? Because all models are wrong. Anyways, with this we could do, since it's 4:30, we're going to go fast through this RCBD example. I just want to show you this. This is what we were seeing before. We could get the marginal means for this RCBD model, right? And see that here we get the standard error, that is 73. And since we assume homogeneous variance, right, that was one of our assumptions, it's going to be the same, and it's a balanced experiment, right? We have same number of observations for everything. We're going to have the same standard error for everything. And here with random effects, we have a standard error of 73. If we fit the same model with fixed effects of the blocks, standard error of 67, a little bit smaller. And that's what we were discussing before, right? No surprise to anyone. The rankings, everything's the same. The point estimates are the same. It only changes the standard error of the mean. And that's because you can go back to a document and think it through again. No huge news with this, right? Example 2 is a split plot experiment. Same data from the package. And now it gets more interesting, because I'm going to give you some background about the experiment. It's a split plot in an RCBD. Factor 1 was different genotypes of oats. And was the whole plot. Okay, so they divided the field into 3, I think, blocks. We'll see later. They divided the field into 3 blocks, and then assigned the genotypes to plots, to each plot. And then within each plot with a same genotype, they assign nitrogen levels. Okay? And that's it. And they want to study the main effects and the interaction between the two. And so, in this case, we have ith genotype, jth nitrogen level, kth block. And this first, the sort of the blueprint, we have the blocks first, right? So the block effect of the kth block. Then the mini blocks. Inside each block, we have a small block, right? And these were actually, I think, 4. So that was the whole plot of the ith treatment in the kth block. Yes? And then the residual. And what we're actually studying is overall mean plus treatment effect, other treatment effects, interaction. Yes? And this translates as overall mean is the intercept 1. All this in R you can write as, the columns are gen star NF. That's nitrogen as factor. I think three levels of nitrogen. And then you see that this is K and this is I in K, right? I nested because this I is nested within this K, right? This whole plot is nested within the block. So there's two ways of specifying that. You could say, like I wrote out there, you can say 1, because we're always affecting the intercept, vertical bar, and then first block, and then nested within that, the whole plot, which is genotype. And the error is just assuming that the family is Gaussian, right? This is the same as doing 1, can you read this? Yes, okay. 1 block plus 1 block genotype combination. Because that's what you're doing, right? It's the block and the block genotype combination. These two are the same. These two, sorry. And so I wish I could show you the data a little bit more, but there's not so much time. So let's see what we get here. This is getting more interesting. Okay, can you read now? Yes. So what do we have here? This is the model we fit, and you see that we have 3 variances, right? We have the variance of the residual, that is epsilon. When we write out this, we should also write epsilon ijk is normal, it means 0, and here sigma hat is 12.15, right? 12.5 squared, right? That's our estimate for the variance. The variance of the blocks, U was 13. And the variance for the whole plots was 9.4. Does that make sense? And you see that we had, this is also telling us we had 6 blocks. 6 blocks and 18 combinations of genotype block, because we have 6 times 3, right? Because we have 6 blocks, like these 3 more, and then inside here we have 3 nitrogen levels, which are, genotypes, which are the whole plots. Otherwise, yeah, these tests you get in R for free are kind of misleading, really, because it's a test if something is different to 0 or not, whether you want it or not. It's a two-tailed test if something's different to 0 or not. Sometimes it's useful. Sometimes it's really not informative at all. Like that the overall mean is different to 0 is kind of obvious, right? If this was a control and you were seeing the difference between the control and the genotypes, it might be more helpful, but yeah. We could, do ANOVA. I want to get to the other examples. Do we have any questions on the split plot model? Also, I checked everything before I came here. So I know these models work because I checked them. It's not that we have to go without checking our model fits. But this all seems fine. No problems yet, right? Any questions? The next example is a repeated measures example. More fun. And the other one is a repeated measures with subsampling. Okay. So this third example, Jordan, do you want to talk about it? This is the temperature data. Do you want to explain it yourself?
Yeah, so this was an experiment where different dietary treatments were fed to pigs. Those pigs have been challenged with LPS, which creates an immune system simulation. One of the characteristic changes is the change in body temperature. So data were collected to look at that change in body temperature over time in response to some different dietary treatments.
Thank you. Well, for that, the design here, this is, you don't have these data, so here you're just gonna have to trust me. First of all, what I'm doing here, this is the more, I wanted to get to these examples because I think they are the most fun. But if you have any questions about the other examples, just let me know. So this is, I try to make it as simple as possible. This is only creating the different columns as factor, right? Factor is it's like a categorical variable with an order, a specific order, right? Character in R doesn't have a specific order. Examples for character could be names, not really ordered, names of countries, names of states. Examples of factor, low, medium, high, 1, 2, 3, 4, but when they are categorical, right? They have a specific order that we should follow. And sometimes it's just easier to have as factor. So, the design, I'm going to show you here, was a completely randomized design, right? So we didn't have, you correct me if I'm wrong, but we didn't have any dependence in the sense of blocks whatsoever. So it was a CRD. We had 5 different treatments. And time as well can be considered a treatment, right? Because we were following it. We know that the background knowledge is that temperature, which is the response is temperature. We know that temperature changes through time after its treatment, so that's something we want to study. That's actually our time is actually our the element we want to analyze, right? If that changes the temperature, and at 5 different points in time. This was measured. And we were following the same experimental unit, right? So that makes this a repeated measures example. So let's go here. Treatment effect, we can recycle a lot of this. Treatment effect, time effect, treatment by time interaction. Do we have block effect? No. Do we have, what would this be? Whole plot effect? No, if we don't have blocks, right? We have the errors, and we want to model the correlation in time. Again, we could model this as a G side or R side, and I can show you how that code would look like. Let's model it G side because it's the way we're learning so far, just adding it in the linear predictor so we don't mess up the whole learning thing. But this would be very similar to the residual, right? Yeah, so it's ith treatment, jth time, kth pen, right? Repetition. This is going to change. And now we can say that this U, I'm going to recycle this. The only thing I should add is that we have 5 points in time, right? So I'm going to add here. But essentially, we're estimating this. We're saying that the observation at time 1 is similar to the observation at time 2, but a little bit less similar. Time 3, a little bit less similar, right? And what we're estimating is the sigma squared U and this rho, right? We're going to look for those in our functions. You'll agree with this model? This is, again, this is called R1 covariance function, and this we're going to say this is time. And this could be pen or pig because in this case there's no subsampling. We could say pen because it's your experimental unit, okay?
That means it's nested in pen. I don't think so. But
Yeah. Good thanks for a question. This vertical bar usually divides This is a little bit different though. In random effects, on the left-hand side, you have the fixed effects, so what the random effects are, or the random factors are affecting, which is usually an intercept. Like in the case of a random block, we have one bar block. And if block is nested within year, for example, we would have year block, right? In this case, it's similar, but on the left-hand side, you have whatever is correlated, right? For example, time, it's the most common thing. And on the right-hand side, you have what individual or what unit is being measured repeatedly. Pen, individual, like when we have a human study, it could be the ID of that person. Does that make sense?
for repeated measures.
For repeated measures, yeah. Usually here, it's where you have time, or it would be some measure of, I think, space. I would read the documentation. This is much more standard than this.
Okay. And the R1 function?
R1 function means that this is a correlation structure. If not, you could say, okay, all the components in the various covariance matrix of this random effect are different. And so you would have 1, 1, 1, 2, 1, 3, 1, 4. But you would be estimating a bunch of different sigmas. And sometimes it's really not that complicated. You can model it as something that decreases with time. The covariance decreases with time, which is why we use very often the R1 covariance structure, and so on. Does this make sense? And I want to show you two things. First of all, I'm pretty sure this won't run, or at least it didn't in my computer. See, model convergence problem, non-positive definite, Hessian matrix, trouble, right? Happily, in this case, a lot of times remember that REML estimation was an iterative process, and sometimes with infinite time you would reach convergence, but some defaults in the computer are set for it to stop if it doesn't find a solution, because it could be running for the eternity, right? Sometimes if you change the magnitude of your response variable, that is more friendly to the algorithm that is looking for those solutions, and that's already that helps you to get a model that actually converges. In the end, as long as you back transform it to whatever you want to interpret, units are invented by us, right? It's not that anything will have more sense than anything else. Units are man-made. So in this case, for example, I switch from temperature in Celsius to Fahrenheit, and you see this warning message, right? If we use Fahrenheit instead of Celsius, it says that the R1 is not meaningful, so apparently this whole thing wasn't really necessary. When you look at the not meaningful means not statistically significant. Sometimes it's not, but it's still, you see a better residual plot, for example, and it's better to keep it. So it's good to not be so test dependent sometimes. And I also wanted to show you how you go about fitting a model with repeated measures. It seems like the R1 structure could be or not be there. Because what we are adding here, we're not only estimating the variance, this sigma squared u, we're also estimating this rho, right? This rho parameter. If we simulate these residuals, we're going to talk about this on Monday. This is also to be used with your own criteria, to be interpreted with your own criteria. What I wanted to show you is this. This is the model we fit. Okay, I'm gonna double check on this. I don't know why it's, yeah, this is giving me, I might have stored the data weirdly. what we're seeing, this is the variance component for this residual, right? And then for the pig or the pen, right, this part here, the repeated measures, we got this for the variance and this for rho, right? Which is, this is kind of odd. I'll double check this later today. But this would be, in general, how you would go about modeling a repeated measures study. Does that make sense? Okay. Then if you go, you can estimate the marginal means. Where actually, if we did an ANOVA on this, we would see that only time is really changing anything. The treatments don't seem to have an effect on time, right? And we can see this with the marginal means and see that they are all about the same. Within each time, they are all about the same, right? Questions? Think today I didn't estimate time as well. We might do the last example on Monday. That's basically the same thing, repeated measures, but now instead of following only one pig through time, we follow, I think, 3, right? The concept is the same, but we have one extra variance component because it's also including that variability among pigs from the same pen, among subsamples, right? And it's something we want to account for to avoid being overconfident about our results, for example. Because if not, if we don't account for that subsampling, we could say, okay, we have a sample size that's 3 times as big as our sample size, meaning number of true replications. That would be 3 times as large as our real number of replications, if we don't account for subsampling. But I do want to dedicate some more time to that, and I know it's Friday. So if you guys don't have any other questions, I will set you free. I will upload this here in the R code page under day 2 and in class code it's going to be. Questions, comments, concerns? Okay. Have a great weekend.